सो लास्ट सेशन में हमने गोल्गी एपरेटस तक डिस्कस किया था. नाउ इन दिस सेशन विल डिस्कस द फर्दर मोर इन द टॉपिक. नाउ द नेक्स्ट सेल ऑर्गेनेल इज lysosome. Now the lysosomes are membrane bound vesicle structure and formed by the process of packaging in Golgi apparatus, right? Now these vesicles, they are rich in all type of hydrolytic enzymes, and these hydrolytic enzymes, they are capable of digesting biomolecules such as carbohydrates, protein, lipid and nucleic acid. In this diagram you can see lysosome as a membrane bound vesicle containing hydrolytic enzymes, and lysosomes are also known as suicide bags of cell. Now, so vacuole is a membrane bound space found in the cytoplasm. It contains water, sap, excretory products and other material which are not useful for the cell. Now vacuole is a single membrane bound structure, and this membrane is called as tonoplast. In plant cell, the vacuole can occupy up to 90% of the volume of the cell, right? Or we can say that in plant there is a large central vacuole is present, and in plants the tonoplast also facilitates the transport of number of ions and other materials against the concentration gradient. So here transport of ions takes place, right? Now in amoeba, a contractile vacuole is present which is important for osmoregulation and excretion. So here in this diagram of an amoeba you can see there are presence of contractile vacuole, and in many cell as in protist, food vacuole are formed by engulfing the food particles. So mitochondria, unless they are not stained, they are not visible under the microscope. Number of mitochondria per cell depends upon physiological activity of the cell. Typically mitochondria is a sausage shape. In this diagram of mitochondria we can see that it is a sausage shape structure. Each mitochondrion is a double membrane bound structure. You can see that there are two membrane, inner membrane and an outer membrane, and there is a intermembrane space between the two, right? The inner membrane dividing its lumen into two aqueous compartments, the outer and the inner. The inner compartment is filled with dense homogeneous substance called matrix. Now in this diagram we can see that the is filled with matrix, right? And the outer membrane forms the continuous lining boundary of the organelle. So we can see the outer membrane is the continuous boundary of the cell organelle, right? Now inner membrane has a number of foldings which we called as cistern, and the purpose of cistern is to increase the surface area. As we know that mitochondria are the site of aerobic respiration, and they produce cellular energy in the form of molecules we called as ATP or adenosine triphosphate. So hence they are known as the powerhouse of cell. So we can see that inner membrane has these foldings which we call them cistern, and they increase the surface area, right? Mitochondria is One of the cell organelle which has its own a single circular DNA, a few RNA molecules, and 70S ribosome required for the synthesis of proteins. Mitochondria divide by fission process. Now, plastids found in all plant cell and in euglenoids. They bear some specific pigments which impart color to the plant. Now, based on these pigments, we can classify them into three categories, that is chloroplast, chromoplast, and leucoplast. Chloroplasts contain chlorophyll and carotenoids pigment which are responsible for trapping light for the process of photosynthesis. In the case of chromoplast, flat soluble carotenoids pigment like carotene, xanthophyll are present. And the last one, leucoplast, are colorless plastid of various shape, sizes, and they store nutrients. And this is a structure of plastid. Now moving on, majority of chloroplast of green plants are found in mesophyll cell of the leaves. Now, like mitochondria, chloroplast also double membrane layer. The inner chloroplast membrane is relatively less permeable. Now we can see that it is also a bimembranous structure. Now the space limited by the inner membrane of the chloroplast is called stroma. We can see this whole place inside the inner layer is known as stroma. A number of organized flattened membranous sacs like thylakoids, they are arranged in stroma. Now you can see that there are sac-like structures which are numerous. These structures, they are called as thylakoids. There are flat membranous tubules called stroma lemmae connecting the thylakoids of different grana. Right, you can see that there is this lamellae connecting the grana. There is one, there's two, and these both are connected by lamellae. The stroma of the chloroplast, they contain enzyme required for the synthesis of carbohydrates and protein. The chloroplast also contains circular DNA molecules and ribosomes. The ribosome of chloroplast were smaller of 70S and the cytoplasmic ribosome of 80S. Now, ribosomes. Ribosome, they are basically the granular structures and they are discovered by George Palade. They are composed of ribonucleic acid and proteins and they are not surrounded by any membrane. Eukaryote ribosome is of 80S whereas prokaryote ribosome are 70S. Now we have already discussed that the ribosome has two subunit, one larger unit and one smaller subunit. Now the two subunit of 80S ribosomes are 60S plus 40S, whereas the 70S ribosome have 50S larger subunit and 30S smaller subunit. So this S is Sweatbox unit and it stands for the sedimentation coefficient, which is a measure of a density and the size of the unit. Now you can see the comparative difference between the 80S and the 70S ribosomes. That 80S ribosome is composed of two subunits, 60S larger subunit and 40S smaller subunit, whereas 70S composed of 50S larger subunit and 30S smaller subunit. Now the cytoskeletons. There is an elaborate network of filamentous and proteaceous structure consist of microtubules, microfilaments and intermediate filaments which are present in the cytoplasm, and these structures, or we can say these filamentous and proteaceous structures, collectively referred as the cytoskeleton, and they involved in functions such as mechanical support. They provide motility and maintenance of the shape of cell. So you can see that diagrammatic representation of microtubules. This is microfilaments and these are intermediate filaments look like. Now cilia and flagella. So basically cilia are short hair-like projection, whereas flagella is a long and whip-like structure. Here you can see a flagella is a long whip-like structure. Now cilia are numerous per cell, whereas flagella is typically one or a few per cell. Cilia are shorter, whereas flagella is longer in length. Now cilia exhibit a wave-like motion, whereas flagella exhibits a propeller-like motion. Now in this diagram we can see flagella exhibiting a propeller-like motion, whereas cilia is exhibiting a wave motion. Right? Now the arrangement of microtubules in cilia and flagella are same, that of 9+2 arrangement. Now if you can see clearly, look at the diagram, there is an arrangement of 9+2, that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 on the peripheral and 1 and 2 on the central. So this type of arrangement of microtubules is known as 9+2 arrangement, which is found in cilia as well as flagella. Right? Now cilia moves in a synchronized rhythmic motion, whereas flagella moves in independent wave-like manner. Now the cilia is found in respiratory tracts, fallopian tube and in the case of paramecium, whereas flagella found in sperm cell and in euglena. Now we will discuss centrosome and centrioles. Now centrosome are a membrane-less organelle that serve as a main microtubule organizing center in the cell, and whereas centrioles are the cylindrical structure made up of microtubules. Right? Centrosome contains a pair of centriole surrounded by a dense protein matrix. We can see that centrosome, they consist a pair of centriole surrounded by a dense protein structure. Right? Now centrosome function is to organize microtubules, aids in mitotic spindle formation and regulate the cell cycle. Whereas the function of centriole, they are the structural component of centrosome and helping formation of basal bodies for cilia and flagella. Usually a singular centrosome is present, whereas two centrioles per centrosome are present. We'll discuss nucleus. So nucleus was discovered by Robert Brown, right? Electron microscopy of nucleus revealed that it consists of two parallel membranes and a space between them which we called as perinuclear space. The outer membrane remains continuous with the endoplasmic reticulum and also bear ribosomes on it. Now in this diagrammatic representation of cell nucleus, we can see that there are two membranes and space between those membranes, and we can see the presence of minute ribosomes on the nuclear envelope, and there are also minute pores which are present on the cell nucleus surface called as nuclear pore. The nucleus matrix, or we can say the nucleoplasm, they contain nucleolus, which you can see here that this matrix is nucleoplasm, and this structure is called as nucleolus, right? It is a spherical structure which is present in the cytoplasm. Now the content of nucleus is continuous with the rest of the nucleoplasm, as it is not a membrane bound structure. So it is a site of active ribosomal RNA synthesis. Now we will discuss about a chromosome. So basically we can say that chromosome is a condensed form of genetic material, or we can say the chromatid contains DNA and some basic proteins which we will discuss in later sessions, called histone protein and some non-histone proteins and RNA as well. A single human cell has approximately 2-meter long thread of DNA. Now every chromosome essentially has a primary constructor or the centromere, on the side of which a disc-shaped structure, or we can say kinetochores, are present. Now here you can see the centrosome, and here the kinetochores are present. Now centromere holds the two chromatid of the chromosome, right? We can see that there is a shorter arm and a longer arm, and is held together by the large central centrosome. Now based upon the position of the centrosome, we can classify chromosome in 4 types. Now first is metameric chromosome, in which middle centromere is forming two equal arms. Now in this diagram we can see that both of the arms, they are of equal distance and centromere is present at the center. Now in case of submetacentric chromosome, the centromere is slightly above from the middle of the chromosome, creating a shorter arm and one longer arm. Now in case of acrocentric chromosome, the centromere is situated close to the end, forming one extremely short and one extremely long arm. Now when the centromere is present on the telomeric chromosome, we can say that it is a terminal centrosome, and such type of chromosome is known as telocentric chromosome. Now sometimes few chromosomes have a non-staining secondary constriction as at a constant location, that gives the appearance of a small fragments which we called as satellites or secondary constrictions.